there. Welcome back to the deep dive. Today, we're going to be taking a closer look at something that might be lurking in your mouth right now. Streptococcus mutans. Specifically, you wanted to learn more about its cell wall and how it's structured.
Right.
Don't worry, you don't need a microscope for this deep dive. We've got some research articles ready, including a fascinating one from Nature Communications that breaks it all down.
It's a good thing we're zooming in, because this tiny bacterium is a big deal when it comes to tooth decay.
Yeah.
You see, the cell wall acts like armor, protecting it and helping it survive.
Okay.
If we can understand this armor, we might just find ways to disarm it.
Okay, so for anyone who needs a quick recap, Streptococcus mutans is this bacterium that lives in our mouths.
Right.
And it's one of the main culprits behind those pesky cavities.
Yeah.
And the cell wall we're talking about, is it like a solid shell around the bacterium?
Not exactly. Think of it more like a chain-link fence.
Okay.
Streptococcus mutans has this unique structure called a rhamnose backbone,
Okay.
made of a sugar called rhamnose.
Hmm.
Arranged in repeating units, kind of like the links in that fence.
Okay.
And just like a fence needs support.
Yeah.
This backbone has side chains attached to it.
Okay.
These side chains are different molecules that give the cell wall its unique properties.
So it's not just a simple barrier. There's a lot more going on structurally.
Right.
It's starting to sound pretty complex.
Oh, it definitely gets more interesting. Now, in the most common type of S mutants found in humans, what we call serotype C,
Okay.
the main type of rhamnose-based carbohydrate in the cell wall is called serotype C carbohydrate, or SCC.
Okay.
Think of SCC as the specific type of chain-link fence this serotype uses for its armor.
Hold on, I need a quick clarification.
Sure.
What exactly does serotype mean? I'm not sure I've come across that term before.
Good catch. Serotypes are basically variations within a species of bacteria.
Okay.
They have slightly different characteristics, kind of like different breeds of dogs.
Oh, okay.
So even though they're all Streptococcus mutants, different serotypes can have slightly different structures in the cell walls, and this can affect how they behave.
Ah, that makes sense.
Yeah.
So back to this SCC fence. Why is it so important for S mutants?
Well, without SCC, the bacterium wouldn't be able to survive.
Wow.
It also plays a big role in how it forms biofilms.
Okay.
You see, bacteria aren't always loners. They love to team up and form these sticky communities called biofilms. Think of it like bacterial cities where they can hang out and thrive.
Right.
And for S mutants, those biofilms are key to causing cavities.
Okay, so biofilms are bad news for our teeth.
Yes.
And this SCC stuff helps those biofilms form. Is that why we're focusing on it?
You got it.
Okay.
Now, remember those side chains we talked about, the ones that attach to the rhamnose backbone?
Yeah.
Well, researchers have recently discovered something fascinating about them.
Okay.
It turns out that SCC has two main types of glucose, or sugar side chains.
So our chain link fence has different kinds of barbed wire on it, I guess? I'm trying to keep up with this analogy.
Exactly. It's a great way to visualize it.
Okay.
The first and most common is alpha glucose.
Okay.
It attaches to a specific spot on the rhamnose backbone.
Mm-hmm.
Think of this as the main type of barbed wire.
Okay.
But here's the twist.
Okay.
There's another modification, glycerol phosphate, or GRO-PP, that could be added on top of that alpha glucose.
So we have the rhamnose backbone, the alpha glucose side chain, and then the cereally on top of that.
Right.
It's like adding extra spikes to the barbed wire, making it even more formidable.
Exactly.
But you mentioned there's another type of side chain.
Right. The other type, though, less common, is beta glucose.
Mm-hmm.
It attaches to different points on the rhamnose backbone.
Okay.
You can think of this as a different kind of barbed wire, perhaps with a different shape or material.
Right.
And what's really intriguing is that even these seemingly minor differences can have a big impact on how the bacterium behaves.
Okay. I'm seeing how these accessories on the fence can really change things up. But how do these side chains actually get built and attached? It seems like a pretty complicated process.
And you'd be right. This is where enzymes come into play.
Okay.
Enzymes are basically tiny workers within the cell that speed up chemical reactions.
Yeah.
Each enzyme has a very specific job.
Mm-hmm.
In this case, think of them as specialized construction crews, each responsible for attaching a specific type of side chain to that rhamnose backbone.
I'm picturing a well-coordinated construction site with different teams of enzymes working together to build this elaborate cell wall structure.
Right.
So who are the key players in this construction crew?
Well, the researchers identified a few key enzymes.
Okay.
CCN and CCM are a dynamic duo.
All right.
They work together to attach those major alpha-glucose side chains.
Okay.
Think of them as the experts in installing the main type of barbed wire.
Got it.
Then we have SCCP and CCQ. They specialize in attaching the minor beta-glucose side chains.
Okay.
They're like the specialists who work with that alternative kind of barbed wire.
So we've got these specialized teams, each handling a specific type of side chain.
Yes.
What about that extra layer of spikes, the GRO-P? Who's in charge of that?
Ah, yes. That would be CCH, our enhancement specialist.
Okay.
This enzyme is responsible for attaching GRO-P to the major side chains.
Okay.
Think of CCH as the crew that comes in and adds those extra spikes, making that barbed wire even more effective.
So we have our chain link fence, different types of barbed wire, and even specialized crews to add extra spikes.
Right.
It's quite an elaborate defense system.
It really is. And what's fascinating is that even small changes to the system can have a big impact on the bacteria.
Okay.
For example, the researchers found that when they deleted the genes responsible for making CCM or CQ, those bacteria ended up with irregularly shaped cells.
Wait, so removing those minor side chain installers messed with the entire shape of the bacteria?
Exactly. It suggests that those seemingly minor side chains actually play a role in maintaining the structural integrity of the cell wall, and maybe even in how the bacteria divide.
Wow, so it's not just about defense. Those side chains are important for the bacteria's overall structure and function.
Exactly.
This is getting more and more interesting, but we're just scratching the surface here, right? There's still more to this story.
Oh, absolutely. In part 2, we'll delve deeper into what happens when other members of our enzyme construction crew go missing.
Ooh.
And we'll start to see why all this matters for you, especially when it comes to keeping those cavities at bay.
Can't wait. I'll see you then.
Sounds good.
Welcome back to the deep dive. We're continuing our microscopic exploration of the Streptococcus mutans cell wall, that intricate fence that protects this cavity-causing bacteria. Last time we met the construction crew, the enzymes responsible for attaching those all-important sugar side chains.
Right.
But I'm dying to know what happens when you disrupt those skilled workers. It's like pulling a key player off a construction project. Chaos. Right?
Well, imagine this. You're building that chain link fence, but suddenly the team installing the main barbed wire goes missing. What happens? It's not just about aesthetics. The fence loses a lot of its protective power. And that's exactly what the researchers found when they messed with the genes responsible for making CCN, SECM, or SECH. Remember, those are the enzymes in charge of attaching the alpha-glucose side chain and that extra spiky layer of GRO-P.
Okay, so they removed those key players from the construction site.
Right.
What were the consequences for S mutants? Did it just fall apart?
Not quite, but it definitely wasn't business as usual. The researchers found that those mutants, the ones lacking those key enzymes, were terrible at forming biofilms.
Right.
And if you recall from part 1, biofilms are like bacterial fortresses, those sticky layers that make it harder to remove them from our teeth.
Hold on, this is huge. Are you saying that without those specific side chains and GRO-PS mutants struggles to build those strongholds that lead to cavities?
Exactly. It highlights just how important those modifications are for the bacterium's survival, and importantly, its ability to cause harm. It's like taking away their building materials, leaving them vulnerable.
This is giving me hope. If we can target those modifications or the enzymes that create them, maybe we can actually prevent cavities from forming in the first place. But wait, how did the researchers even figure all of this out? It's not like they can just watch these tiny processes happening in real time.
They used some pretty impressive detective work.
Yeah.
To get to the bottom of this, they used a combination of powerful techniques.
Okay.
One of those is mass spectrometry.
Okay.
Imagine a supersensitive scale that can weigh molecules with incredible precision. That's what mass spectrometry does.
Wow.
It allowed the researchers to identify the different components of the cell wall, like those glucose and GRO-P modifications, with amazing accuracy.
So they could tell exactly what building blocks were present in the cell wall.
Yeah.
That's pretty amazing. But knowing the ingredients is only part of the story, right? You need to know how they all fit together.
You're absolutely right. That's where another powerful tool comes in. Nuclear magnetic resonance spectroscopy, or NMR for short. This technique is like taking an MRI of a molecule. It allows researchers to determine the precise structure of those modifications and how they're connected to the rhamnose backbone.
So mass spectrometry tells us what, and NMR tells us how.
Exactly.
It's like having both the parts list and the assembly instructions for the S-mutant cell wall.
A perfect analogy.
I love it.
And by combining these techniques with genetic manipulations like deleting those enzyme genes, they could pinpoint the specific roles of each player in the construction process. It's like removing a worker from the assembly line and observing the consequences.
It's like a microscopic game of Clue. CCN in the cell wall with the alpha-glucose.
Right.
But I'm curious, of all those modifications, GROP seemed to be a real VIP in this story. Why is it so special?
GROP plays a critical role in the formation of those stubborn biofilms. Remember, these are bacterial communities that are notoriously difficult to remove. They're a major contributor to tooth decay.
And the researchers found that no matter what sugar it was attached to, GROP was essential for biofilm formation.
Right. They even replaced the glucose side chains with a completely different sugar, GLCNAC, one found in another bacterium called Streptococcus pyogenes.
Wow.
And you know what? Even in this foreign environment, GROP still enabled the bacteria to form biofilms.
Wow. That's pretty convincing evidence that GROP is a key player in this whole biofilm game. But
Yes,
what does that mean for us? Why should we care so much about this tiny molecule?
because understanding GROP's role can be the key to unlocking new strategies for fighting tooth decay.
Mm-hmm.
What if we could develop treatments that specifically target GROP, disrupting its function and preventing those biofilms from forming in the first place?
Oh, okay, that's getting me excited. It's like finding the Achilles heel of S mutants.
Yeah.
But are scientists actually looking into this? Is there any real potential for GROP targeting treatments?
There's definitely a lot of interest in this area. In fact, researchers are exploring a few different approaches.
Right.
Some are focusing on developing drugs that could directly block GROP's activity, kind of like jamming the signal that tells the bacteria to stick together.
That's fascinating. So instead of killing the bacteria outright, we're trying to disrupt its ability to form those pesky biofilms.
Right?
But are there other strategies being explored?
Absolutely. Another approach involves targeting the enzymes responsible for attaching GROP to the cell wall. Remember CCH, our enhancement specialist. If we can find a way to block its activity, we might be able to prevent GROP from getting at it in the first place.
So it's like stopping the construction crew from delivering those extra spikes for the barbed wire?
Exactly.
I'm really seeing how understanding the structure of this cell wall is opening up new avenues for fighting cavities.
Right.
But I'm sure there's even more to this story, right?
You bet. In Part 3, we'll delve into the exciting world of vaccine development and explore how researchers are using their knowledge of the S mutant's cell wall to develop potential vaccines for preventing cavities. Stay tuned. It's going to be a fascinating ride.
Can't wait. I'll see you then.
Sounds good.
Welcome back to the Deep Dive. We've been on quite a journey exploring the microscopic world of Streptococcus mutans and its intricate cell wall. From the chain link fence of the ramnose backbone to those spiky GROP modifications, it's been a fascinating deep dive.
Right.
And now for the grand finale, vaccines.
Yes.
I'm really curious to hear how researchers are using this knowledge to develop potential vaccines against cavities.
It's a really exciting area of research.
Yeah?
And it all boils down to this: our immune system is incredibly powerful, but it needs to know what to target.
Right.
Remember those side chains on the cell wall?
Yeah.
They act like identity tags for the bacteria.
Right. It's how our immune system recognizes those invaders and mounts an attack.
Exactly. So researchers are exploring ways to use those side chains and even GROP itself
Okay.
as the basis for vaccines.
Okay.
The idea is to train our immune system to recognize these specific components of S mutants so that it can fight them off before they can even establish themselves in our mouths.
That's brilliant. It's like giving our immune system a set of blueprints so it knows exactly what to look for and how to neutralize the threat. So are there any promising vaccine candidates in the works?
There are a few different approaches being explored.
Right.
One strategy involves using synthetic versions of those cell wall side chains as the basis for a vaccine.
Okay.
Basically, scientists create artificial replicas of those identity tags and introduce them into the body.
Okay.
This triggers an immune response, so when the real S mutant shows up,
Yeah.
the immune system is ready to attack.
It's like showing our immune system a wanted poster with a detailed description of the culprit. So if S mutants tries to sneak in, it's immediately recognized and dealt with.
Right.
That's incredible. But what about Group P? Can it also be used in a vaccine?
Researchers are definitely looking into that as well.
Okay.
Rupi is such a critical player in biofilm formation that it makes a very tempting target. The idea is that a GROP-based vaccine could trigger the production of antibodies that specifically bind to GROP, interfering with its ability to help those bacteria stick together.
Yeah.
No more bacterial fortresses, no more cavities.
I'm really blown away by the potential of this research. A vaccine against cavities would be a game changer for dental health worldwide.
It would.
But I imagine there are still some challenges and hurdles to overcome before we see a cavity vaccine at our dentist's office.
Of course, vaccine development is a complex and lengthy process.
Right.
One of the biggest challenges is ensuring that the vaccine is both safe and effective.
Yeah.
It needs to trigger a robust immune response, without causing any harmful side effects.
That makes sense. Safety is always paramount, especially when we're talking about something that would be given to a large population, potentially even children.
Yeah.
Are there other challenges researchers are grappling with?
Another hurdle is figuring out the optimal way to deliver the vaccine.
Okay.
Should it be an injection, a nasal spray, or even a mouthwash? Each method has its pros and cons, and researchers are carefully evaluating the best approach for a cavity vaccine.
So it's not just about developing the vaccine itself, it's also about finding the best way to get it to work where it needs to be.
Right.
It's a reminder that even the smallest details can make a big difference in science. But despite these challenges, it sounds like there's real hope for a future where cavities are a thing of the past.
Absolutely. The research we've discussed today is a testament to the incredible progress being made in understanding the biology of S mutants.
Yeah.
And this knowledge is paving the way for new and innovative strategies for preventing tooth decay. It's a truly exciting time to be in the field of dental research.
I couldn't agree more. This has been such an eye-opening deep dive. I'm walking away with a newfound appreciation for the complexity of this tiny bacterium and a sense of optimism about the potential for a future free from cavities.
I agree.
A huge thank you to you, our expert, for guiding us through this fascinating journey into the microscopic world of Streptococcus mutans. And to you, our listener, thank you for joining us on this deep dive. We hope you've learned something new and are as excited as we are about the future of dental science. Until next time, keep exploring, keep learning, and keep those pearly whites healthy.